So, welcome everyone. We have Dr. Saurav Shukla, and he'll be giving us a guest lecture today. So, Dr. Saurav Shukla is currently a joint postdoctoral research fellow at the University of Washington and PNNL. He received his PhD from chemical engineering at UIUC, and he also has graduated from chemical engineering at IIT, Indian Institute of Technology, and he's worked on a lot of different publications and topics. He's very passionate about protein and drug discovery, and also passionate about mentorship. And so he's here to talk to us about studying dynamics of proteins and drug interactions with machine learning. So let's welcome everyone. Let's welcome Dr. Saurabh to for our guest lecture, and Saurabh, over to you.
Thanks, Professor Karthik, for the kind introduction. And hello, I'll share my screen with you all. Okay, can you see my screen? Okay. Yeah, so thanks, Professor Karthik, for the invite. And it's my pleasure to talk to you about some of my work on using machine learning on large scale simulations of proteins and understanding their dynamics and the drug interactions. So I realize that it's a fairly new topic to the students of electrical engineering department. So I will try to be very detailed in explaining things and the background. But please feel free to interrupt me and talk to me if you have any questions or clarifications. Let's make it a discussion instead of a lecture. So there'll be a lot of new things, and I hope I am able to communicate those to you. So let's start very fundamentally about life, how life works. And life means all the organisms we see: humans, animals, plants, even small bacteriums or the viruses. So basic building block of life is the cell. And you see animation here, and you must have heard about DNA.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Yeah. So this is a very condensed version of how life works. So in every organism, the information is contained in the DNA. So DNA is sort of the storage mechanism which transfers the information from one generation to another. So we contain the DNA in our body, which comes from our mother and father, and which is combined, which is inside us. And that DNA is translated into the RNA. RNA is a protein, which is basically RNA is the entity which is the middle entity, and then eventually that RNA is translated to the protein, and the proteins are the powerhouses of our body and of life. They do all the work. So if we are seeing, if we are hearing, our limbs are moving, our brain is functioning, the proteins are doing all the work. If we have a sense of touch, we have a sense of smell, if we have a sense of taste, there are proteins which are recognizing the stimulus from the environment, and they are processed in our brain, and the brain also is working because of the protein. So proteins are the building block of life, and today in today's lecture we are going to understand how we can understand the mechanism of these proteins. So here I'll give you the example of two proteins which I also work on. These are called molecular motors. So here you see that these are like real smallest motors of the universe, where the protein you can see here, the kinesin protein, is walking just like a human, one step at a time on these roads which are there in every cell of our body. So in our brain also these roads are present, these motor proteins are present, and they are constantly walking from one place to another. So for example, if you want to basically operate your toe, then the signal is sent from the spinal cord to the toe, and the signaling molecules are carried by these proteins. So this sac, which you see, this is called organelle, which is transported by this protein from one place to another. So these are very important proteins, and in a similar fashion, these proteins are made by different instructions which are already encoded in our DNA, which we have gotten from inheritance from our parents. And these proteins have specific shape. Every protein has a different shape. So the motor which I showed in the last slide, that also had a very different shape, which enabled it to walk on these roads. And these are very complicated proteins, which are basically designed by the nature, by the billions of years of evolution in our or in the universal organisms. So let's go into the fundamentals of the proteins, how they work. So protein has a sequence. It's basically 1D array, if you will. If I talk in terms of computer science, it's a 1D array of amino acids. And there are 20 different kinds of amino acids present in living beings. So you can think about it as a 1D array where each element could be 1 to 20. And based on this sequence and the how these array is designed, each protein has a different structure, and because of the structure it has a different function. So here the phenomena of protein folding is explained. So initially the protein is translated or made in terms of this 1D array, which is a chain, and by the phenomena of protein folding, it folds itself based on the charges, based on how big these amino acids are in a particular shape, and this phenomena is called protein folding. And this is a very fundamental phenomena, which is very important in the life processes. And if the protein folds or misfolds, that's when the disease happens. So the disease like cancer is caused by a protein called Src kinase. And the cancer is spurred because it misfolds itself. And when it misfolds, it basically gives constantly signal to the cell to grow and grow. And that's why tumor happen, because the cells are constantly dividing and dividing and dividing. There is no checkpoint. And that is the protein folding problem. And that is why I'm giving you a example, because that's why it is so critically important to understand how these proteins fold and dynamically interact with themselves as well as with other proteins.
Quick question.
Yeah.
So do we know why proteins misfold, like you mentioned? Is there like another cause for it, or is it still not understood? Understood.
Yeah, so proteins misfold, primary reason is mutation. So when you think about a disease like cancer, there are some tests, genetic tests like BRCA1 and think BRCA1, BRCA2. So there, so let's say there is a chain, it is supposed to be healthy in a particular order, but one or two elements of that chain are swapped, let's say, and that's why we say we have genetic predisposition of cancer. If let's say in our family somebody has cancer, or our grandparents or our aunt, because the genes in those our family tree are mutated, and we inherit those genes, and because of that mutation the protein misfolds, because the sequence has some errors into it, and that is why these diseases happen. So this is one of the primary reason of the chain is itself faulty because of our genetic reasons. Yeah, so one of the ways to study these proteins, so there are two ways to study these protein. One is computational way and one is experimental way. So today I will not be talking about the experimental way of studying this protein, but I will be talking about how do we study these proteins computationally? How do we study the motion of these proteins computationally? And here I am showing an example of a protein which is being simulated by by a method called molecular dynamics simulation. So molecular dynamics simulation says that we provide the sequence of these amino acids to the computer, and based on the charges and the size and other properties of these amino acids of the chain, we tell the computer to calculate the forces on these amino acids and let it run so that it basically eventually folds into a structure it is supposed to fold. So here I am showing an example of the complete simulation of a protein, where it will start with a chain, which every protein starts in our body, and it will fold into a characteristic structure where it functions. So this is a movie which is made by large scale computer simulations on supercomputers. So you see these chains interacting with each other, and they are making these interactions. They are folding into a particular shape. The C terminus is this, which is coming to. Yeah, so this is the final folded structure of this protein, and you can see how these different interactions formed. And we saw this movie in the time scale of a second, but this phenomena takes less than a millisecond. So it's really fast. So if you want to study the proteins using a microscope, the time resolution needs to be extremely fast, and we need to be able to see extremely short time scales. So this protein will be 5 nanometer, and we don't have technology today to see these proteins folding at such a high time resolution, at such a high spatial resolution. And that is where these molecular dynamic simulations come in, where we can study this folding phenomena and how the proteins misfold as well. We can basically simulate the proteins with mutations, and we can simulate the healthy proteins to see how these two differ from each other. So now I'll just go in the mathematical details of these molecular dynamic simulations. So what happens?
Saurabh, quick question.
Yes.
So given a sequence of amino acids in a protein and given a particular gene structure, is the ultimate protein folding structure uh, like unique, or are there like multiple stable configurations for a protein to fold into?
Yeah, that's a great question, I think, and I answer is exactly on this slide. Uh, so, uh, so when proteins fold, there are millions of conformations which are possible, right? And it is defined by this energy funnel-shaped energy landscape. So initially, these chains lie on this top periphery of this funnel. Then there are several local minimas present all over the place. So mathematically speaking, if we want to understand the native most stable structure of the protein, let's see, if we start from this chain and we do the calculations, we might reach into this minima. Can you see my cursor? Okay, yeah, so we reach into this minima, and mathematically speaking, when we reach into one local minimum, it is extremely difficult to get out of that energy barrier and go into the global minima. So that's a mathematical challenge when we run these simulations. And as you can see, there are multiple minimas possible. These are called intermediate states where protein reside for some time, but most of the conformations of the proteins reside in this global minima, which is the most stable structure. And if some of you are familiar with this protein structure prediction, it has been a very, very difficult problem to solve computationally, coming up from sequence to the structure. And there have been multiple recent advancements like AlphaFold, Rosetta Fold, which is just our campus development, which now very accurately predict protein structure from these sequences, which have just recently come out from in 2022 using deep learning. Yes, Blake.
So are these local, or can you hear me?
Yes, please. Yes.
Okay. Are these local minima, are those like misfolded proteins that kind of stay misfolded, or are they sort of just in a semi-stable state, and then when they finally reach the global minima, they're in their most stable state?
Yeah. So these are intermediate states. That's a great question. These are not misfolded states. These are the intermediate states. So it's basically a equilibrium ensemble, right? There are millions of proteins being produced in a cell. Most of them, 95% would be in this completely rightly folded state. These are intermediate states where a protein resides for some time, and that it will eventually transform to this most stable state. And there is, believe it or not, our cells or bodies, such a complex and exquisite machinery. Our cells have machinery to detect the misfolded proteins and recycle them. So we have mechanisms in place where misfolded proteins are detected and recycled, are broken down. Continuously it is happening in our cell. But these are intermediate states. So and so I'll start this slide with a quote, very famous quote: "Everything that living things do can be understood in terms of jiggling and wiggling of atoms." And this is such an important quote because when protein folds, and as you saw in the previous movie also, the atoms of these amino acids were jiggling and wiggling, and because of that jiggle and wiggle, that random motion, thermal motion, they come in contact with each other, they feel attractions and repulsions, and that is how, through that jiggling and wiggling, they basically collapse into a very stable structure. And how we do these simulations is that we give these computer structure of the protein. It can be a chain, or it can be an unstable structure, and there are 4 or 5 terms which we provide based on each atom. So we basically define the position of each atom. We put them in a water box. So basically there is a box of water molecules around the protein, because every living things every living process are carried in the water buffer in our cells. And there are 4 energy terms which are assigned. One is bond energy. So if there is a carbon which is bonded to oxygen, that there is a bond energy there. There is an angular energy. So there are 2 bonds and there is an angle between them. There are some forces involved in that. There is electrostatic force. So if there is some atom which is negatively charged, which interacts with the positively charged atom, that they will interact with each other, they will basically attract. And there are van der Waals attractions as well, which make these things possible. So what we do is we assign the position of every atom, which could be thousands, 50,000 atoms, a million atoms, or 100,000 atoms. So basically somebody recently concluded the large scale simulation of the COVID proteins, which was millions of atoms and which takes enormous amount of supercomputing time. And one thing which I should mention, the time scale of these simulations is very, very slow. So each time scale is femtosecond, which is 10 to the power minus 15 second each step. So the steps are, positions are defined. So at every step, the forces are calculated on every atom. And based on that, after a time scale, the position is calculated. Based on that force, the next time step, the position is calculated. And if you can imagine, we calculate the forces on 50,000 atoms of the protein and all the atoms of the water which is surrounding it. So let's say I am simulating 100,000 protein atom system. So I calculate forces on 100,000 atoms, and then every step I calculate the next position of that atom. And that is why if I simulate a very small protein, to simulate 1 millisecond dynamics of that protein, it takes like 6 months runtime on supercomputer with hundreds of GPUs. So that's how slow this protein simulation process is. And you can imagine how much data we generate in studying these proteins. And that's where the machine learning and data analytics thing comes, while after running these simulations, when we have that large terabytes and terabytes of data, we use machine learning and specifically unsupervised learning to make sense of these protein molecules dynamics.
One more question, actually. What is the objective function for this problem? Like, I understand that it is something where any of these local minimums are something where no amount of jiggling can get you any changes, right? But interesting, I mean, the interesting question is, how do you quantify it in an objective, or is that too complicated to describe?
So basically, in the simulations, what we are doing is we are observing the protein in an unbiased fashion, how it behaves. So when we run the simulations, we basically start the simulations and protein itself behaves in a manner it is supposed to behave based on its structure, how the forces are being produced, what atoms are closer to each other, and how it is going. And then now there can be multiple cases. Let's say we have large scale data of a protein, how a protein is behaving. Then there are multiple things we can study. We can study how a protein is folding, one. Second, there are multiple active or inactive states of a protein. So let's say the cancer proteins are kinase. When it is in its active conformation, it tells the cells to divide. When it is in its inactive conformation, it basically tells cells to hold on, not to divide. So in a perfectly healthy human being, this process is regulated. Sometimes it's active, sometimes it's inactive. But in a faulty protein, it is always active. It's always telling the cells to divide, divide, divide. And that's where we have uncontrolled growth, and we have like tumors in the specific organs of the body. Third application could be objective, could be to study the drug interaction. So let's say SAR kinase, there is a drug which was recently discovered, which is called Gleevec. It's called imatinib or Gleevec. So what that drug does is it basically finds that protein, binds to that protein in a conformation which regulates that protein. So most of the drug which we take are small molecules which go and bind to a particular protein to regulate it, to make it right, to correct its folding, its misfolded structure to the right structure. And that is how, so the third application could be to study drug binding. So how the drug goes and binds to the protein and how it affects the structure of the protein, how it corrects or miscorrects or misfolds the structure of the protein. So these are the objectives, different mathematically speaking, we are solving this equation, the potential equation, and we basically let the program run and see how the system is behaving.
Sorry, follow-up question.
Sure.
In terms of the protein, becoming active or inactive, is the information to do that contained in the protein, or is it getting a signal from elsewhere to do that?
So it depends on the protein. So most of the cases, the active and inactive information is inside the structure of the protein. Some parts of the protein determine whether it is active or inactive. So they are sometimes in a different structure. So the structure of the protein is called conformation. So sometimes it is a different structure, which is an active structure, and a different structure, which is inactive structure. For example, our body, right? If I raise my hand, it's active conformation. If I just put down my hand, it's inactive conformation. The parts of the protein are the same, but they're in different positions. And that's how the active and inactive is determined. And protein is doing it all the time, but most of the cases in a healthy human being, it is happening in a regulated fashion. But when it does it unregulatedly, then the disease happens. And that's where we need drugs to basically stabilize that structure in that regulated structure of the protein. Yeah. So here today, what I'll do is I'll basically give you a case study. So this is a study on plant protein. So in plants also, we talked about human proteins. In plants also, the same thing happens, DNA to RNA to protein, and the proteins do all the work. So here there is something in plant produce a hormone called ABA, abscisic acid. And so this ABA hormone, so hormones are like drugs. So the drugs we take, they basically go and find its protein and they stabilize it. So the hormones are also like proteins, are like drugs, which basically goes and binds to the protein they are supposed to bind. So ABA is a plant hormone which basically binds to its corresponding protein. So the hormone proteins are called receptors because they receive these hormones and bind to these hormones. And this is important in providing drought resistance to plants. So let's imagine a scenario, a drought prone area. If you basically use this hormone on the agriculture land, the plants don't dry even if there is no rain for months. So it's very important in ensuring food security of the future, and that's why it is important to study it. So our objective function while studying these protein is that how does this plant hormone binds to its protein, binds to its structure. And that's where we did the molecular dynamics simulations. And here you see different proteins which are found in plant, and you see different, and here in the structure you can see there is a binding pocket. There is a place where this hormone goes and binds. That's right now it's sitting here in that binding pocket. And important conformational entities of this structure is these loops. So this loop, and they have different names to it. And the very important loop is this, which is called CL2. And you see the blue structure, it is kind of a closed conformation on the top of this hormone. And this light blue is some open conformation of this loop. So it turns out the closed structure is the active conformation. That's where this protein works really. And this open conformation is the inactive structure when the protein doesn't work. So we wanted to study how this protein, the hormone binds to this protein, as well as how this opens and closes and how that activation happens. So these are the steps of molecular dynamics simulation. We initialize the structure of the protein. So we basically put this protein in a water box and put the hormone outside the protein. and then we calculate the forces from the particle positions. So if a atom is sitting and there are bunch of amino acids around it, then based on what is around it, it will experience certain force, and based on that we calculate its position. So we solve the Newton's equation, which I just showed in a couple of slides below, and then we write the next position, and then we loop it over and over again. The time step is 1 femtosecond. So you can imagine going to millisecond as like millions of steps, billions of steps. It's extremely slow. We have to run it for 6 months on like hundreds of GPUs to get millisecond scale dynamics of these proteins. So I'll show you a movie which basically we get after running these large scale simulations. So this work was done when I was at University of Illinois at Urbana-Champaign. We had a supercomputer on campus. So we were lucky to get supercomputing resources to run these large scale simulations. So initially the hormone is outside and you see the protein is jiggling, and it is trying to find its binding position where it wants to go, and it's searching. And now you see that it has found the opening, the binding pocket. It went inside, and then you will see that now it has found a stable position inside this protein, and you can see this loop. Now it's in its inactive conformation, which is open, and at the end you will see that this loop will close on the top of the protein, where this protein will become active. Right now it's an open conformation, and this hormone which is sitting inside is trying to activate this protein and make it close. Oh, it came outside. So sometimes the hormones basically go inside and come outside also, because there are millions of structures possible, and that also happens sometimes. All right, so now the machine learning part comes. You can imagine the data set is the position of 50,000 atoms at each time step. And time steps could be millions of time steps. We can't possibly use this data for analysis of what is happening. So what we do is we select the most important feature. So we featureize these trajectories to apply the machine learning models. So what is the most important feature here? So if you look at the structure, there could be multiple features. There could be distances between the two atoms. So one feature could be a distance of this hormone molecule, ABA molecule, from the binding pocket where it is supposed to be. So when it is outside that feature, that distance will be more. When it is sitting in its binding position, that distance from binding pocket will be less. So how do we select the most important feature? One is explain the phenomena the best. So this comes from the biological understanding, that comes from the expertise. So I know that the distance of this ABA molecule from the binding pocket is the very important feature, because it describes the binding of that ABA to this receptor, minimizes overfitting, minimizes bias, and fast and robust. So these are some criteria to select these features. And in these simulations, the features could be the distance of these molecule, distance of these loops from each other, which explains whether the loop is closed or open. It could be the conformation change of this entire global protein structure, feature called RMST, root mean square deviation. So basically we calculate the square distance of each atom from its previous position, and then we add it, and then we take a root. So that gives us the global change in the entire protein structure. So we can use software called Visual Molecular Dynamics, VMD, to see these proteins and manipulate these proteins, which I will not go into the detail today. But now what we do is we classify these structures, these billions of structures we get, into common clusters. So we perform unsupervised learning to classify these protein structures in clusters which are very similar to each other. So what one analysis we do is we featureize these protein structures in multiple features. So basically these are the distances of different amino acids with each other. We calculate the most important features. So basically features which basically describe the phenomena the best, the importance value of these features. We do the feature selection initially. And here I have plotted, I don't know if you can see or not, I have plotted two features and plotted the conformations here. So APO is the inactive structure, that's a scientific term. HOLO is the active structure, that's a scientific term. And you can see these are the two features where these APO structures which are inactive are clustered in one position and the active structures are clustered in one position. So there is a decision boundary, linear decision boundary, which you can draw to basically segregate active and inactive structures. And there are different sets of, and here also there are two different features which basically segregate active and inactive structures. So the way to study these structures is to featureize them in important features. And this is the same plot which was described below, where active and inactive structures are being segregated based on two different features, and the features are distances between two amino acids which are at important locations in the protein. So what we did in this project was we also can study the time scale. So time scale progression of how this drug is binding, this ABA is binding. So initially the distance of this drug from the binding pocket was very large. So initially it started from here, and then as time progressed, you can see when the this ABA is bound to this binding position, the distance of ABA from the binding pocket becomes very, very low, which gives us the indication that the drug has bound. And we can plot these conformations on a heat map, where it is these, each dot on this plot represents a protein structure. And these are two features. This is the distance of the ABA from the binding pocket. This is the feature representing whether the loop is closed or open. And you can see that here I have shown you the structure also. When the ABA is away from the binding pocket, these are the structures. When the ABA is bound to the binding pocket, these are the structures. And you can see there is a energy minima here where most of the structures lie, and this is a very stable structure of the protein. And by plotting these histograms of different structures, we can identify the local minima of these protein structures. So basically, to analyze this data set, we perform the k-means clustering. So here you can see one cluster where you can see all the proteins which are similar to each other are clustered together. So we use three features to cluster these protein molecules. RMSD of loop 1. So there are two loops we use. So loop this loop and that loop. And because these are important in describing the function of the protein and the distance of ABA from binding pocket. So based on these three features, we k-means cluster these proteins. And clustering is important because we can't deal with millions of structures, but we can cluster these proteins in similar structures and assume that this cluster represents one state of the protein. And these clusters will give us the intermediate structures. The question of Blake, the intermediate structures, we could also get what are the intermediate structures of this entire progression of this protein. Yes.
So quick question, all of this is based on the simulation, right? That
Yeah.
we are So how accurate are the simulations in, like, how do we know about, because it's all based on the 6 months GPU's base simulation that you mentioned. Like, do we know it's very accurate, or do we know it's pretty close to?
It's extremely accurate actually, which is very, very amazing. So as you will see that in this project, we actually started from inactive conformation. So we knew inactive conformation of the protein from experimental studies. Techniques like X-ray crystallography or cryo-EM can predict or can know the protein conformation. But it gives only one static structure. We also know the crystal structure of bound and active conformation. So when we started this project, we only knew two structures, what is active, what is inactive. But through this simulation, we started from inactive without any information of the active structure. We basically recapitulated the entire binding pathway of this ABA molecule, and we obtained the active structure from the simulation. So we had some structure to verify our results, and we verified it that we did actually get the active structure from starting from the inactive structure. So these are extremely accurate simulations, and these simulations are used in pharma industry to find new drugs. Because let's say there are drug candidates, you can put these drug candidates inside the binding pocket of the proteins, and you can see how that protein is behaving just by doing these simulations and not doing these experimental screens, which cost billions of dollars. And that's why the drug discoveries traditionally has been so slow. This is actually employed in pharma industry to do the real drug discovery. So very accurate simulations. In fact, 3 years ago, the Nobel Prize was given who invented these simulations, Michael Levitt, to basically have invented such a technique which can produce such authentic results without doing any experiments. So very accurate. Yes, so basically this is something which we used, which is called elbow method for number of clusters. So in k-means clustering, the question is how many clusters do we provide the algorithm? 10, 100, 1,000, a million? Because we have billions of structures. How do we actually calculate? How do we know how many clusters to make? So we did something called elbow method, and which is a very general method to calculate the optimal number of clusters in a k-means algorithm, where we start from different number of clusters and we calculate the error of k-means classification method. And basically, generally what happens is the error generally decreases and later it plateaus. And we knew that we could calculate, we could select a number of cluster which basically at the beginning of this plateau, because as we move along, as we increase the number of clusters, we are just splitting two very similar, we are splitting a cluster which is very similar with each other, and there is no point of splitting the clusters more. So this is something which can be used to calculate the number of clusters which are optimal for any unsupervised classification problem. So then what we did was we made these clusters of protein. So when we took that data and we classified or clustered these protein states in let's say 100 states, and each dot here in this network represent a cluster of these proteins, and these connections give us the transition probability of this structure from one to another. So what is the probability of transition from going from this cluster to this cluster? We also calculated the probability. So we employed a method called Markov state models, which is a kinetic method where we can calculate the probability of protein going from one structure to another. And here a plot is generated where we can basically reproduce how the protein would have behaved based on that model on like thousands of microsecond of time. So we didn't actually simulate this much time, But based on this Markov state model, we can calculate how this protein is going to behave. So here, if you focus on the bottom graph, it's a distance from binding pocket to the ABA. You can, and these low values are when the ABA is bound. And you can see the ABA doesn't remain bound all the time. It unbinds, floats around in the water, again binds, floats around the water, and binds. So it's not a very stable phenomena. And from these simulations, we can see what is actually happening physiologically, which is not possible to do with any experimental technique which is currently available. And here we have this. Yes, Karthik, you have a question?
Yeah, sorry. So in terms of this clustering, right, we're looking at a single protein, but different structures during the simulation and clustering those intermediate structures, right?
Yes, so it's the same protein and we have millions of structures. Since we can't study millions, we cluster them in like 100 structures, which are distinct from each other. And
So
then
does this help us understand other proteins as well? Like, I'm just curious, like through the study of clustering and understanding structures of a single protein, where else can we apply this, the clustering method? Like what's the big motivation for doing the clustering?
So the motivation is to simplify our analysis. One. Second is to calculate these transition pathways from one state to another. So these clusters represent an intermediate state of binding of that protein, of protein landscape. And to understand how protein traverses from one state to another. So on this graph, I have shown it is an inactive state. It starts from here, the protein, and this is active state here. It goes to this active state. And there are multiple pathways possible from going from inactive to active state. And these arrows, the width of arrows represent the probability of that pathway. So the protein goes from this pathway, which is the most prominent pathway, but there are other pathways possible. For example, this pathway goes from this to this, here, here, and here. But the probability of protein traversing from this pathway to this pathway is very low. And this is a very complex network of transition from one state to another. And using that clustering is the first step to understand these different pathways and to describe the process with the complexity which it happens really in the real life. That's the motivation.
So understanding the pathways, does it help with, let's say, drug discovery better? Like, I'm just curious, like, is it understanding for our, like, to have a deeper sense of how things work? Or does it actually help us speed through the process of drug discovery as well, like for other proteins?
Yeah, it certainly does, because sometimes what happens is, let's say the inactive structure of the protein, if we want to basically develop a drug, then we need to find a drug which perfectly fits the protein. There could be a drug which fits the intermediate structure of the protein, but not the inactive structure. So if we only know the structure of the inactive protein, the development of the drug is not possible. Sometimes we need to match the drug to the intermediate structure. And to match that drug to the intermediate structure, we need to know what that intermediate structure is. And that is why it is important to understand the pathway, because so that we have, for the same protein, we have multiple conformations that to target that drug to. So that is why it is important. Yes. Yeah, so basically the idea of this is, so here we only use the unsupervised learning, but that was so crucial to understand this entire pathway. Otherwise, we would not have been able to recapitulate this entire pathway and calculate these transitions of these networks from one state to another. So conclusion of this entire project is that we started from this inactive structure, we had this active structure. So this was known experimentally. And outcome was this. that we started from the drug outside, and then we know this entire pathway where the drug found this binding pocket. It stabilizes this, stabilizes in this binding pocket, and then you will see this loop will close. And the drug is actually attracting this loop to close over on the top. And now this is the stable conformation of the protein. Now the loop is closed. And this is the conformation where the protein is most effective. So yeah, so that's one project. And so let's see if somebody has any questions or want to discuss anything. Yes, please.
On the previous slide, there was a yellow state in the middle of that state graph. Was that state, was that sort of where like the protein kind of found maybe where it was going to go and then realized it was a bad state and sort of backtracked out? Because there's no further arrows going out to a state that it wasn't already visited.
Yeah, that's exactly it is. That's excellent point and a great observation. So this is an unproductive state where it's kind of stuck. So it is supposed to go in a certain orientation. It is stuck in a reverse orientation. And now it can't go anywhere. So it's stuck. And these are called unproductive states. And while making a drug for a protein, we want to avoid these kinds of states, or sort of we want to minimize the occurrence or the probability of these states. So that is why these unproductive states are very important, because some drugs get stuck in these unproductive states, and that's why they are not effective. They even basically make the conformation worse. So that's the reason why these states should be avoided.
Okay, so comparing that to the first graph on the first page, the local minima and the global minima, is that are all these intermediate states the local minimas where it's finding a state? So would that mean this unproductive state would be some sort of extraneous local minima that it gets stuck in and then backs out of, or is it just an inactive state that's just bad?
Yeah, it is a local minima.
Okay.
The local minima, it basically protein sits on this position for a while, and it could be how deep it is. It depends on how stable it is, how long the protein is residing there. So that's the deal with these states. It could be unstable. It could be just like with a blick of a protein, it just goes and then it comes out because it doesn't like the way it is sitting there. But sometimes it can be stable. It can sit there for a very long time.
Oh, wow.
It doesn't help the protein.
Cool. Thank you.
Yeah. Yes, please. Yes, James, do you have any question? I can't hear you if you are saying anything. Is there a question? Yeah, but
James,
the
do you
ground
have a
truth, that
question?
is X-ray crystallography steady state confirmation, right? Is there any ground truth dynamic state confirmation data available? So there is dynamic ground truth. There is no dynamic ground truth which is available from experimental technologies, because the getting that structure is itself a large scale effort, because these are so small. Seeing the amino acids is so small. These ground truth data is also obtained from millions of these protein structures, different conformations, and then mathematical models are applied to these millions of conformations to get to this ground truth, the static structure. And dynamics is just not possible with current state of the art. The ground truth, so only thing which we can do is we run these simulations and compare our model to this ground truth structure which is available. If we can recapitulate it without guiding the simulations, then we know that this is right. If we can't, then there is no way to do it. And one more thing which I would like to comment on, there are some proteins which are very difficult to crystallize. That is why it is very difficult to get their structure. And that's where the simulations also come into play, where Experimentally, it is not possible to get the structure. The computationally, we can possibly get the structure of that protein. Any other questions or comments?
So essentially, if we want to make a faulty protein work or make it change the state through a drug, we simulate the pathway from active to inactive and identify the key transition points and see if the drug is able to hit any of them or all of them. If they don't hit the early ones, then we discard it. Is there something like that? Like, because it's a sequence. Like the most probabilistic pathway, if you're not able to hit the early transitions, the drug is not able to reach, modify the protein to that point, then you say it's not worth going further. Let's not, continue. Does that help with screening drugs in that way? Or
so basically, how this does drug discovery? So you can imagine that for this large scale simulation is not possible for every combination of a drug and a structure. So what people do is they take the static structure, they put the drug molecule in its binding pocket, and then they simulate for some time whether that structure, the drug is stabilizing that conformation or not. And then they can do it for thousands of structures and thousands of drug molecules. And that's how small simulations are run to screen for the drugs. You can't run, for months, the simulations for one drug and one protein. So these simulations are used to get the structures, and then with different combinations, the simulations are run to see what is the effect of the drug, whether the drug is actually stabilizing or destabilizing.
so stabilizing is important, but also getting to that structure is also important, right? Like if the protein is not in that place, and we want the protein to change active to inactive, or inactive to active. So how do you also know if the drug is actually helping the protein get there? Like, is it? I don't know if I'm making sense, but like if I'm already in an inactive state, then I can say I want to be there. Then the drug helps me stabilize that state. But if I want to actually get to an active state and I'm actually active, do we also have a way of measuring that for a new drug?
So only way is to basically run the simulations with the drug. You start with the structure and you see whether it is going to that state or not. And the only thing which we avoid in drug discovery is that we don't put the drug outside. We put the drug inside that binding pocket in the right orientation. So the amount of calculations is exponentially decreased because drug is already there. It doesn't need to find the pocket. It just need to modify the local structure. And then we can see whether it is modifying that local structure in direction of that active structure or not. Does that answer your question? Hello?
Yeah, it does answer.
Yeah. How are we doing with time, Karthik? I think.
I think we're good. We can go for another 30 minutes. Yeah, we do have time if you want. Yeah.
Yeah, so this was one project, and there is one more project which I can briefly describe, which basically is more in the domain of drug discovery. It's not discovery per se, but it's basically drug classification. So here we use neural nets for basically seeing how drugs are different from agrochemicals. So agrochemicals are the drugs which are given to plants. So pesticides, insecticides, the drugs which basically spur the growth of the plants and the vegetables. These are agrochemicals. And here the drug I will refer to human drugs. The drug is which is given to human. So there is one problem which now we all as a society are facing, that these agrochemicals, specifically pesticides and insecticides, are having adverse effects to human health, because they are given to agricultural products and the humans consume these products, and then these pesticides, which are supposed to work on plants, they also interfere with human proteins, because you can imagine the proteins. And so more than 95% of the genome of human, the plants, is same. Uh, so proteins also has very high similarity. So it's possible that the drugs which work on plants also interfere with the human proteins. And that is why it is very important to understand how drug structure and the agrochemical structures differ from each other. Yeah, so this, I think, I have given an introduction that the pesticides also have impacted human health in an adverse manner. And this is just a slide say that drug target protein in human body, but also agrochemicals target proteins in plants. Because structurally they are similar. And every drug molecule has a structure. It's a small molecule, and this is the drug which I was describing, which is imatinib, which is an anti-cancer drug, and its commercial name is Gleevec. And you see that it has a structure, it has some rings to it, there are some side chains like the CH3. Some rings are just benzene rings, some rings has a nitrogen in it. So each drug molecule can be featureized in its characteristics. And characteristics can be what is the molecular weight of this drug, how many hydrogen bond donors are there, how many heavy atoms are there, what is the polar surface area. Surface area is if you basically make the structure of the drug, how much is the area of this drug, of the surface. If you imagine these atoms as spheres, how much is the area of this drug? And this is an agrochemical where we can featureize this molecule also. And if you look at these human drugs and these agrochemicals, structurally they are like made up of the same entities, some bunch of rings, bunch of carbon and nitrogen. And we wondered whether we can classify them in drugs and agrochemicals. So there are ways to featureize these drugs based on these properties. And we started to featureize these drugs, and there are different properties which are desired in a drug, like potency, bioavailability, non-toxicity, non-reactivity, solubility, which is very important. Potency is how potent is this drug, how much doses one needs for it to work. Bioavailability is if you put it in the bloodstream, how likely it is go to the organ of the interest where it is actually being used. Whether it is non-toxic or not, non-reactive, it doesn't need to, it should not react to the proteins and make them inactive. How soluble it is. If the drug is not soluble, then you can't just take it. It will just go through the excretion system of the body and the body will not receive it. And these properties of the drug are dependent on the structure of the drug. How lipophilicity, hydrogen bond donors are there, how many rings are there, what is the size, what is the double bonds. And there is a very crude rule which was proposed by a scientist called Lipinski, that for a drug to be effective, it needs to have a molar weight less than 500, polar surface area needs to be less than 140, and some bunch of the rules which now we know that are very crude descriptors of these drugs. And some drugs which are very effective lie outside of the domain of this rule as well. So now if we basically compare the drugs and agrochemicals, so I'm making the bunch of plots, the histograms of agrochemicals and drugs. So agrochemicals is in the green and the drugs are in the blue. So these are basically distributions of thousands of agrochemicals and thousands of drugs. Here I have plotted the polar surface area, hydrogen bond acceptor atoms, hydrogen bond donors should be here, octane water partition coefficient. And here in all these properties, you can see that agrochemicals and drugs are not awfully different from each other. And from just doing the histograms, it's very difficult to classify these to class of drugs, which is agrochemicals and drugs. So then what we need is we need more features, of course. We can't just go by these 5 rules by Lipinski. And we need more detailed and complicated models to classify these drugs. So these are the same thing which I have plotted here. And it's clear that histogram method doesn't work. And what we did was we used neural nets. So basically we featurized these drug molecules and looked at how these neural nets are able to classify these molecules in drugs and agrochemicals. And I'm sure the neural nets would have, yes.
Yeah, sorry, I just wanted to get a quick motivation. Is the motivation to differentiate agrochemicals and drugs that we don't want drugs to be like agrochemicals, like because of unintended or unwanted effects of agrochemicals on plants? So maybe same thing from drugs on human proteins. Is that like it aids or helps navigate drug discovery and weeds out drugs that are not useful based on this association? Is that like?
Yeah, so the idea is that agrochemicals which are awfully similar to human drugs should be avoided. So it basically is more intervention in the agrochemical discovery pipeline than to human drug discovery pipeline. So when somebody develops a new agrochemical, let's say to spur the growth of tomato, they can screen that agrochemical in this model to see how it is going to affect the human health. So that's the overall motivation behind it. If it is too similar to the human drugs, then probably it should not be approved because it is going to have adverse effect on human health.
Got it.
So that's a more and like that's overall picture. Can we make a model to segregate these two class of agrochemical and drugs so that we can classify them based on their features?
Or maybe we can just go organic and avoid agrochemicals.
That's true as well. Yeah, so I will not go into the details of this function because I am sure the neural nets have already been covered. But these are the details of the model. We had like 195 features as like how big it is, how many number of rings it is, how many double bonds, how many single bonds, things like that. And these are some of the details of neural nets, which I don't think I need to go into the detail, which might have been covered already in the course. But the conclusion of the project was that we obtained 95% accuracy of the segregation of drugs from agrochemical. It was a simple feed-forward neural network with 5-6 layers. And that itself was able to classify these drug molecules from the agrochemical molecules. And I also applied different machine learning techniques like decision trees. And at the end, basically neural net was the one which basically was most effective in classifying these drugs from agrochemicals. But the point here was this slide was made for students of chemical engineering. But I think this is very intuitive to the people in ECE that every model, machine learning model has different accuracy based on the problem. And it is very important to try different models to optimize your accuracy and tweak the model according to your application as well. Yep, so I think that's all from my side. And if anybody has any question, we can on either of the projects we can discuss more as well.
I have a quick logistics question on these accuracies. On the previous slide, you said 95% accuracy of training data. Was that, I'm assuming it was like you trained it on a certain amount of data and then you tested it and validated it on a separate set of data. And that's where the 95% accuracy came from, right? Okay. Cool. Just want to clarify that.
Yeah, it's on the test set.
So for this, is there currently some kind of process that companies have to go through to introduce a new agrochemical, or it's only by human trial?
So right now there is no such pipeline available. This is just for the research purposes. So every time a research does, it goes through bunch of steps before incorporating into the real drug discovery pipeline. So this is very preliminary. This is exploratory work. And as of now, it has not been adopted by anybody because it's very preliminary. But I think it has a potential to go into that pipeline also. And as of now, drug discovery pipeline is very crude. It's set in stone, and it will take a lot of effort to disrupt that pipeline. Many startups are doing work in this direction of incorporating data science and machine learning into the drug discovery pipeline. But the traditional big pharma companies is still that large scale drug screens of experimental drug screens. That's still the state of the art. It will be few years, if not decades, before we incorporate these ML pipelines into the drug discovery pathways.
So another motivation question, like when we look at the effect of agrochemicals on plants and plant proteins specifically, and you said plant proteins have a similar makeup as human proteins. So now the question I kind of have or I'm trying to understand is, when we consume plants that have proteins modified by pesticides, how does that interact with, what we already have in our system, right? Or is it like a long-term effect of it inducing mutations in the proteins that we already have in our body?
So let's take it that way, right? There is a pesticide which we consumed, and there are millions of different types of proteins in our body. And to understand whether that pesticide interacts with some of the protein or not, it's a very complicated problem to solve, because for that we'll have to use that pesticide with different protein particles and see whether it binds to something or not. But it's very likely that some of the pesticides have the structure which is complementary to the protein in the human body as well. And the side effects we see from these pesticides, from some of the pesticides, for example, one of the classic case was DDT, right? It was banned because much later it was discovered that it is interfering with the human bodies. It is harming the human body. So some pesticides whose effects are visible in terms of human health are recognized, but there are certainly pesticides available which are currently being used, which are harming our body in terms of interacting with the human proteins, which we have not discovered yet. We might discover it 10 years later when we see that effect of it on a large population, and then we might realize that, oh, this is bad and let's ban it. But
So,
it's
and then we can and this can be extrapolated from pesticides to even like regular food that may not be healthy, right?
Yeah.
Over long term, it can. So this is about long-term consumption, because I consume once and I don't consume it again, I'm fine, because only proteins at that time in my system get impacted. But then long term, I'm just having more and more of these weird proteins in my body because of interactions, and that's so it's pretty interesting.
Yeah.
So we really don't understand, like pesticides is great, but we really don't understand how, other kinds of food that we may have is actually impacting the makeup of cells and proteins in our body.
Yeah, I mean, we actually don't understand a lot, and this is one way of thinking about whether it impacts human body or not. But human body is also very diverse, right? My body is different from your body, and something might react to my body very differently than which it reacts to your body. And that's where the field of precision medicine and personalized medicine comes, where we study the effect of different things based on the genetic markup or the makeup of me or you, and how different drugs and different treatment regimens are effective on my body versus your body, right? Some cancer patients respond to chemo very well, but some don't, right? And that is the same drug is going for the same type of cancer, but the genetic markup is different for every human, and that's why different drugs react very differently to humans, different individuals. So that's another layer of this entire discussion of understanding.
Any other questions for Saurav?
I have a question related to the first project. The way I understand these things like AlphaFold and stuff is that once it's trained, it kind of makes a simpler model of the protein folding than the traditional one you were talking about. Have you heard of, or are you aware of any kind of machine learning model that has led to making a maybe a better understanding of the process, or more elegant understanding of how physics works or something like that?
So I'm confused about the question. Are you asking about how? Can you please explain your question?
Yeah. So I guess there are two kind of approaches to the protein folding problem. One is to go from the ground up and use the physics simulation to build the protein, and then the other is to train an ML model using basically labeled data or something like that, where you don't give it the intermediate steps and it kind of figures out a model on its own. Do people deconstruct those ML models to understand maybe like a more efficient way to describe the forces in the protein folding problem?
Yeah, so there are two different problems. So molecular dynamic simulations, which I described today, that is non-forced approach, where we just let the system run. It goes where it wants to go. It goes to the most stable minima. And it's a very natural way. That's why it's very slow, but it's very reliable because we're not biasing the system in any manner. So that's one way. Second way is AlphaFold or the Rosetta fold, which just came, where you give a sequence of the protein and it gives you the final structure. And there is a very deep neural network working, which has taken millions of these chains and their corresponding structure in training the algorithm to figure out a new structure from a new sequence, right? Here you have only two data points: one initial chain, final structure. There is no information, intermediate information. There is no dynamics here. It's just simple structure prediction from the chain going boom. So you feed a structure, boom, you feed a chain, it's a linear array, boom, you get a structure based on that model. Intermediate way, how people expedite this situation is people can apply forces in these molecular dynamic simulations. If I have a knowledge a protein is supposed to go in this state, I can actually, so basically one way is I can wait for the drug to bind. Second is I apply the force to drug to go to that binding pocket and see what happens. That's a way to expedite these discovery pathways. And people do that. That's also a legitimate way to, do these calculations, and people do that all the time. So that's one way, other, which I didn't talk about today. You can just bias the system based on your understanding where it's supposed to go. So you can do that as well.
I think maybe also the question was about interpretability, right, of AlphaFold. I don't know if that was also the question. Like, if you give the starting structure and the final structure, and we know that the molecular dynamic simulation is accurate. So somewhere the deep learning model is actually learning how to understand. I mean, it's able to understand how the forces actually come together and change the structure into more stable configurations, right? And I guess maybe the question, I don't know if the question was about, are we able to see some of those things show up in the layers of the AlphaFold or one of the deep learning models? Right?
Yeah, possibly. Actually, that's a very good point. That while the AlphaFold is learning, it might be, let's say, in intermediate layers of the neural net, we might be having these states which are actually there in the protein in the molecular dynamics simulation. So let's say output of each layer might correspond to a different intermediate state. That is possible.
Thank you.
Yeah.
Okay. If any last questions anyone has? Yeah, thank you so much, Dr. Saurav, for joining us today, and getting us more into the protein folding and the protein side of things. And I think it's been very enlightening to go into so much of the depth and details. So yeah, thank you for joining us, and I think this talk is recorded, so we'll have it posted also for more public consumption soon. And yeah, if you have any materials and slides, we'll also share it with students later.
Yeah,
Thank you
thanks
for
so
joining.
much. Thanks for giving me an opportunity to share my work here. And there were many great questions. So thanks for that.
All right. So good.
Yeah, I hope we learned something about how life works. Yeah. Yeah, I was a chemical engineering student, and when I learned first about the proteins and the DNA and the RNA, it's just blew my mind how exquisitely this entire body and the machinery is working. It's just mind-boggling.
Yeah, I mean, and then also when you talk about drug discovery and all of these things, we're actually tinkering with the fundamentals, right? Like, it's amazing that the drugs are going at such a fundamental level to make things work. It's really fascinating.
Yeah.
Okay. So yeah, thank you so much for joining, and we'll maybe see you again soon.
Yeah. Thank you. Thank you, everyone.
All right, guys. I think that's about it for today. We'll stopping the recording.